Hello everyone, I am Dr. Tarun Kumar Patel, Assistant Professor Biotechnology from Sant Guru Ghasidas Government PG College, Kurud, District Dhamtari, Chhattisgarh. In this lecture, we are going to discuss about identification of protein sequence from DNA sequence. This topic is useful for MSc Biotechnology, second semester, paper 5th, Biostatistics, Bioinformatics, and Computers in Biotechnology, Unit 4. Let's see. The translational tool. There are a number of translational tools are available in different websites. These translational tools are basically used for the identification or prediction of protein sequences from query sequence. The query sequence might be DNA or RNA sequence. Some of the important translational tools for conversion of DNA to protein sequences are as follows. Embose transcript from EMBL EBI. Embose transcripts translates nucleic acid sequences to their corresponding peptide sequences. It can translate to the 3 forward and 3 reverse frames and output multiple frame translations at once. Emboss 6-pack. It reads a DNA sequence and outputs the 3 forward and optionally 3 reverse translations in a visual manner. Expert Protein Analysis System or ExPASY translation tool. It is developed by Swiss Institute of Bioinformatics. It is a tool which allows the translation of a nucleotide, DNA or RNA sequence to a protein sequence. Let's see how we can identify protein sequence from a query DNA sequence using TransQ Emboss tool available at EMBL EBI. This is the interface of EMBL EBI sequence translation page. Here you can see the nucleotide sequence translation tools as TransQ Emboss and 6-pack. To select the TransQ Emboss, click on the button Launch TransQ. When you click this button, you will reach to this interface. This is the interface of Emboss TransQ. Here you can see to find out the protein sequence through Emboss TransQ, we have to follow or proceed some steps. The first step is the entry of the input DNA sequence. The DNA sequence or the nucleotide sequences can be manually entered or can be copy pasted, or the file of query sequence can also be uploaded here. When you enter your input sequence, then the step 2 is selection of parameters. 2 parameters can be adjusted as per the requirement of user. The first one is the frame, number of frames, and second one is the codon table. The default value for frame is 1, whereas the default codon table is standard code. Then the step 3, submission of the job. In this step, if you tick a box, a notification will be sent in your mail when the result is available. And finally, click on Submit button to get the result of the analysis. On clicking the Submit button, you will find the result in this manner. Here you can see the sequence of amino acid of the entire DNA sequence. The result can be displayed in color format as follows, or alternatively the result or sequence of amino acid can be downloaded. In this way, you can translate a DNA sequence into a protein sequence using Emboss Transque tool available at EMBL EBI very easily. In addition to this Emboss Transque tool or the other tool we have already described, there are various tools available for the DNA to protein translations. Let's see briefly some of them. The Open Reading Frame Finder tool of ANSI can also be used to find out the protein sequence from a query DNA sequence. The ORF Frame Finder searches for open reading frames in the DNA sequence you enter. The program finally returns the range of each open reading frame along with its protein translation. ORF Finder can be used to search newly sequenced DNA for potentially protein encoding segment, verify predicted protein using newly developed Smart Blast or regular BLASTP. Alternatively, there are some other web links are also available for DNA to protein translation as depicted here, whereas the 6' translation can also be done using the TubeIngen tool or the Russian Molecular Biological Laboratory website or using the tool BioLine. These tools are also very useful for finding out the protein sequences using DNA query sequence. So finally we can say that the protein sequence can be identified from DNA sequences using the translational tools and the other tools available at different websites very easily. We have to follow the procedures as described in these websites, or sometimes some websites also require a specific format for entering the query sequences. Some require the FASTA format for the query sequences, or sometimes they can accept the gene ID. In this manner, anyone can easily identify the protein sequences from DNA sequences.